Okay, so we'll kick off handing over to Steve Hoad, who's heading up the project here at Balbirnie on the plant health side of things at the strategic farm.
Well, good morning, everyone, and really the objective for this particular presentation is to try to make some introduction to the activities that we're doing on the farm, on the strategic farm, and we really want to try to use this as a starting point. So this is just the beginning, really, of this kind of journey, working together, bringing science and practice together, and hopefully through the topics I've just listed there, you'll feel as though we're beginning to cover some of these things today and into the future. And the key thing is going to be getting ideas from this community, particularly the farmers within the community, about the ways that these kinds of meetings and the activities go on these strategic farms. So Chris has made the introduction to Balbirnie Home Farms. So please look at the information that's out there on the AHDB website and on the strategic farm website. I've just flagged up a couple of the links here. So there's a wealth of information out there already, and that will build. So today, I just, as I say, wanted to kind of provoke some ideas and think about the ways we're beginning to look more holistically at the idea of crop health, very much in the crop rotation and the sort of health of the whole kind of rotation and the whole system. So these are some of the things that come to mind for us when we're thinking about working with the farmer-led approach, giving as much support as we can along the way with all the emerging technologies that might be able to support the different approaches. So you can see there's all kinds of factors that we consider when we embark on setting up pieces of work on farms. And we'll try and cover one or two of these things today, but certainly into the future. So what I'll do in a moment, I'll just focus on one of the aspects of plant health regarding the crop nutrition trial at Balbirnie Home Farms. But around that, there's quite a lot of other activity on plant disease, pests, and soils. And Paul will certainly include some aspects of the soils work a little bit later. So I suppose our philosophy is really trying to bring together both the productivity aspect, but also we're trying to bring in the idea of maintaining efficiency, improving the efficiency. So on this scheme, this is actually a scheme we're developing with our students. So I thought it's just a nice illustration of essentially what we want to do for yield, output, or quality. We want to move from left to right as far as we can, and then for efficiency, likewise, we want to move from the bottom to the top as far as we can. So it's bringing all of these things together within our strategic farm systems approach. So we see how far we can go from perhaps the orange, the yellow, and into the green. We're basing quite a lot of our ideas on what's gone before in terms of the way we work on farms, the kind of measurements that we make. And this is just an example of an ADAS publication where they've neatly put together some guidance, some ideas about how particular activities could be or should be carried out on farms of different sizes and different business structure. So I urge you to look at this this guide that is actually freely available online. So we're actually using this to develop the kind of approaches that we take when we're looking at activities, certainly across several fields or across the whole sort of farming business. We want to see what the best ways of capturing information about crops, what the best information is, and and also to try to get a feel for what is going to be the most informative type of measurement program to help the farming business and the farming philosophy. So along with this, we're actually working towards embracing technologies. Now, this is something to come in future, but what we see happening is the things that we do on the grounds as people going out and measuring things, and the way that farmers and agronomists will measure things. We want to actually begin to link this to the emerging technologies, and I think that's going to be the key towards getting things towards what we call sort of more real-time management, or more effective and efficient management. And this is just an illustration of some of the science we're carrying out at the moment, but we'd like to really try to apply this to real kind of farming situations where we're linking grounds with remotely sensed information, and really kind of finding out the best ways to embrace this information. So our approach, and this is focusing very much on just one of the fields that we're working on at Bourbonnier Home Farms, and so this is for the crop nutrition trial. And in this particular scheme, so we've got a field here, almost 14 hectares in size. And what we're doing, we're actually creating zones within the field that are based on sort of three main tram lines that we're monitoring. So 1, 2, and 3 there, and then we're using these zones to try to follow the crop through the season under different management. And we might regard management as being perhaps standard, or a standard practice, compared to perhaps something that's very much a low input system. And then another approach where the farm can make decisions, update decisions, more or less on a day-to-day basis as new information comes in. So this is just an illustration of how this works with our tramline system based on 36 meter tram line, and then when each of the tram lines, so we've got zones in which we can follow the crop through the season. Now this is winter wheat variety LG skyscraper in this particular field, but across the project we've got different crops in different fields across the farm. So these are the different tramline conditions that we're considering in terms of health and nutrition in this part of the work. So as I say, we've got a standard approach in one of the tram lines in terms of the fertilizer, but without fungicide. And then in tram line 2 there, we've got what we might call the standard agronomist managed program on this particular farm in this particular area. And then we've got a third tram line where we're trying to make adjustments according to the weekly or sometimes the daily measurements that we make on farm. Essentially what we're trying to do is to put some value on these particular approaches. How much does it cost to roll out these particular approaches, and what does it mean in terms of the crop, the yield, the quality, and the efficiency of the system? It's not necessarily about yield, it's about other things. It's about margin and sort of longer term benefits for the crop rotation. These are just some of the measurements that we are considering in terms of understanding changes in crop health and physiological condition. So it's quite a list here, but we might go through some of these more detail in the breakout session. So there's quite a lot of sort of physiological measurements, and some things are actually being done already on farm. You know, farmers are embracing some of these particular kinds of measures, looking at things like canopy color, greenness, which is obviously quite widely used already in terms of nutrient management, and we're using other methods such as linking measures of crop ground cover to estimates of crop size, and other new approaches we're taking is more regular tissue or sap analysis, and we're using other measures such as the use of refractometry to look at the physiological condition of the plants. And one of these methods is through what we call the use of the Brix unit system, which is looking at solute or sugar concentration within leaves. So these are some images from much earlier in the year at Burnley. So we're following through measures and photos, looking at change over time, because the key thing in this type of work is to be able to measure as frequently as possible the impact of the decisions that we make. So from this year, which involves quite a lot of very intensive assessment, we'll try to pick the best or the most informative methods for going forward in future years. And that's where you come in as a wider farming and agronomist community in terms of how we might refine these methods, and also perhaps learn from your own experiences. This is just another illustration of the kind of things that we're doing just across the different tram lines. So we're getting these visual representations each time across the different zones, and then what we're doing there is trying to match these to some of the physiological measurements that we make. So we're building up quite a body of data now on the different measures. Now you might say, well, this is very sort of costly and time consuming, but our approach is to try to decide really what is going to be the best, the most informative approach for us going forward. So just to finish in the next couple of minutes, just to show you a couple of the ways that we're pulling data together. So first, the way we're looking at the plant health status is through the nutrient content of leaves and the developing canopy. Now, I don't expect you to see the fine detail here of all the different nutrients, but essentially the horizontal bars represent different nutrients of interest through a SAP analysis test carried out at a commercial lab, as you can see there. So we're following this through different stages so we can actually look at change over time, and we're trying to see how sensitive the system is in terms of the measurements we make and the change we see affected. Now, it's quite dense in terms of the amount of data that we capture, but what we hope to do is to refine this so that we can target particular phases of growth that we believe is more informative for the farmer management. So the blue bars indicate sufficient or excess. The green bars essentially kind of on track, and the red bars flag up where there might be some deficiencies. So nutrients such as manganese, magnesium, and calcium have been ones that we've been trying to rectify where we've had low levels. Just to finish off, we're trying to develop different approaches. Now this setup here is actually in another field where we've been testing some equipment. So it looks quite kind of like a laboratory like in this particular figure. But what we're doing is testing how different approaches work, and this is using the refractometry or the Brix analysis system. So we're just trying to test this to see how meaningful that might be. And certainly David, our host farm manager, is actually using this quite widely on farm, capturing a lot of information, sometimes on a daily basis, so that we can actually put some real meaning to the sugar content, the solute content measures that are made. Now, the actual measure is influenced by the time of day that the measurement is made, or even the weather conditions, because it relates to the physiological activity of the crop. So what we're trying to do over time is to understand how we might best use this particular measure, and how well it might inform us on making decisions, or more effective decisions. So this is just the final slide, and it just shows over a number of dates here how the Brix reading changes. So from early March towards late May, there we've got Brix readings on the y-axis. And you can see that the left-hand figure, this is our standard agronomy. The right-hand figure is our more managed, adjusted agronomy. Now, they're quite similar overall in terms of the level of the Brix reading, and some of that variation on the level of the Brix unit will be to do with the climatic conditions at the time of making the measure. But there's some suggestion that the Brix reading is slightly higher overall in the more kind of adjusted system. But we need to fully validate that and indicate how that relates to other features of the plant's health. So I'm going to finish there. I hope that's been sufficient to provoke some questions going forwards. And hopefully we can take some of these ideas on design approaches, ways forward into the breakout session.
Perfect. Thanks very much for that, Steve. Certainly very, very thought-provoking, and just, you know, running through the season so far at Bulburnie, it's been interesting just to see these results collated and presented so far, which there will be a results session later on in the season for most of the trials. So what we'll do is there'll be a breakout session in 40 minutes, 30 minutes, which you'll get a chance to really dig into Steve if that's the choice, and we can discuss the trial further. And great work that Steve's doing trying to quantify all the great advances that David's been making here at Bulburnie and the testing and adjusted nutrition. It's really quite exciting stuff. So we'll move on now to Paul Hargreaves, and we'll catch up after Paul's presentation. Okay.
I'll just unmute myself there. All right. Good morning, everybody. My name is Paul Hargreaves. I'm really going to talk about soil and soil health today and some of the thinking behind soil health and how that's been implemented in this piece of work at Bulburnie farm, and how that kind of relates to the nutrients and the things that have been measured in the plant as they grow, in the crop as they grow. Just to start off with, I mean, this is quite a basic slide, but just to come back to thinking about soil health, we're thinking about the whole soil. So not just the mineral particles part of it, it's the organic matter, it's the water content, and it's the air as well, the spaces, the porosity of the soil. So it's trying to think more than just the chemical aspects of the soil. And this really is trying to illustrate what we're really thinking about when we're thinking about soil and trying to improve soil health. In the past, there's been a major focus or impetus on looking at the chemical aspects of the soil, making sure that the plant nutrients or the nutrients that the plants need are there in the right amounts at the right time for the plant to use. So that's been a lot of the focus of soil analysis. Where with soil health, we're trying to also think about the physical aspects of the soil and how the texture and the structure of the soil play their part in allowing these nutrients to be taken up by the crops, and also how this happens through the soil biology and the microbial populations within the soil, and how these have an influence on how the chemicals in the soil, the nutrients that you've applied, are being made available to the plants that are growing there. So it's not just the soil chemistry, it's when we're thinking about soil health, it's the physical aspects and the soil biology as well, and trying to assess these and trying to show how these are important. And basics are important in all of this. Your soil texture is important, understanding what soil texture you've got, whether it's a light sandy soil or a heavy clay soil, can all have an effect on the nutrient uptakes, the growth, and how these affect the take-up of the nutrients that you're applying to the soil. So understanding what soil texture you've got to start off with is quite fundamental in understanding how these interact and the overall choices for your soil health. And this is important. This is just to illustrate this here with excess winter rainfall along the bottom axis here against nitrate that's been leached from the soil through winter rainfall or excessive winter rainfall, and the different soil types here. Some of them, the silty soil holds on to the nitrate a lot more, even with excess winter rainfall, than a lighter, sandier or loamy sand soil. So again, this is important on understanding your texture, about what will happen during certain parts of the year, and the nutrients that are available may not be available or be lost due to different soil textures. So this is important to start to understand this whole process. And then soil structure, this is different to texture. This is how the particles of the soil bind together to form the building blocks within the soil aggregates. And this is important because this is where the roots of the plant anchor to the soil and where they get the nutrients from. It also needs gaps and pores within those blocks of the soil. This is an important part of the soil structure, as these allow water to drain through, but also retains some with the micropores and some of the organic components of the soil, where moisture is retained within the soil as well. So this is an important part of the soil structure. So again, as I said, with water retention, and also it allows air into the soil as well. Roots need gas exchange, oxygen to grow, as well as the microbial population within the soil all need air to get in there to work at its maximum efficiency. So these are ideas that we're looking at, and we're using the VES system, which is taking, as you can see here, a block of soil and pulling that apart to look and identify the kind of soil structure that you've got, and grading that from a score of 1, which is a good soil structure, through to a poor soil structure of a score of 5, where you've got very platy material. There's roots are having difficulty moving through the soil, and so you're not getting that efficient uptake of nutrients and growth for the plants. And again, a more compacted soil structure also reduces the biodiversity of the microbes. And in some cases, if it moves from aerobic conditions, where there's plenty of air getting in there, to anaerobic conditions, where you've got standing water or water being held within the soil profile, then this changes the whole dynamics of the microbial population, and you can move from a very efficient soil microbial populations to very inefficient and poor turnover of nutrients. And you can tell this sometimes if your soil becomes is quite has a sour smell, or smells of rotten eggs, then it's the microbial populations aren't working as efficiently as they should do. And this is just an illustration of how soil structure is important. The bottom dotted line here shows a more compacted soil, whereas the blue solid line shows a less compacted soil. And it's crop yield up the side here against applied nitrogen along the bottom. And you can see with 50 kilograms per hectare of nitrogen applied, then you get a much better yield from the less the soil with better structure and less compaction. And also this really illustrates that if you're going for looking for the same yield from the crop, from the more compacted soils, then you've more or less got to put on twice the amount of nitrogen to achieve the same yield as the less compacted soils. So not only is this affecting yield, it's potentially increasing costs on farm as well. Now, we just thought I'd just focus slightly on some of the soil analysis that's been done and the importance of these. General analysis for soils of pH, pK and Mg, and also in some cases people think about trace elements as well, and also mineralizable nitrogen to see what nitrogen is available to the crop there. And pH is important, as we can see here. This is where we get the soil where it goes from acidic soil, so a low pH value, to alkaline, a high pH value, and where we're really aiming to get pH values around 2.6 to 6.2 to 6.4 here. And this shows that means that the availability of the major nutrients that you're applying to your crops or the crops need to grow are available here, where it's just slightly acidic. So pH of the soil is really important. Also, with some of the trace elements, they're also more readily available at this pH as well. So that's why your pH is important. And crops have an optimum growth for pH. This has been defined through science and research over the years, but you do see poor growth outside of those optimum pH range, and there's general things to look for within the plant as it grows. But again, confirming this through a pH test is really important. And just again, finally thinking about what you add to improve the soil pH is it's important to think about the neutralising value of the lime or products that you put on there. They're not all the same. Some of them have got much higher neutralising values than the other, and also some of them can add some of the other nutrients as you improve the pH. You can also help improve the magnesium if that's lacking in your soils. And just thinking about what's been happening on the farm here, we've been looking at pHs in a more detailed manner using a grid system across the field. So instead of just taking a W of samples and bulking those together to get, say, a mean value, in this case for this field would come out at about 5.2. You've got areas within the field that are high in pH, and you've got areas of the field that are low in pH. And basically adding your lime as a variable rate, knowing where these areas of the field are, you should be able to help save money in lime, and also try and bring the whole field up to a level, a pH level that is good for consistent growth across the whole of the field. And these are just some examples of some result sheets, not particularly from this particular farm, but just thinking about what we're measuring here. It's the importance of the extractable phosphorus, extractable potassium, extractable magnesium, and extractable calcium. These are important words to know on these kind of sheets, because this is the amount available to the crop itself. What that crop should be able to extract from the soil as it grows. And this is important because if you're looking just at total P, there could be a lot more P within the soil, but this is locked up and not available readily to the plant. It may become available over time, but it's not available to the growing plant itself. And also seeing where these are within the bands of levels within the soil that show what's optimum for crop growth, and if you need to add any more of these. Because with the soil test, basically you're trying to see what conditions are like prior to as the crop starts growing. So you're trying to create the most optimum growing conditions prior to that. What were the tissue testing and tissue sampling comes in, that shows how the crop is reacting to that and what it may be deficient in. It might give you some indications of some kind of trace elements that might be deficient, and that what you may be able to rectify that through the growing season. And this is just a more detailed results here that's showing that there's other aspects of the soil analysis that can be looked at, from the C/N ratio to show this is all soil characteristics that really look more closely about the microbial population and how well they're doing, what the conditions are like for them, and in turn the turnover then of nutrients through that population. And this just brings us on to think about the organic matter within the soil. Scottish soils should have high levels of organic matter, between 5 and 10%. So there should be a measurable amounts of organic matter within your soils. And soil organic matter is important because it helps retain moisture within the soil. It provides sites for microbial activity. It provides slow releases of some of the nutrients that we see and we know that the crops need, for especially the N, P and K aspects of it. However, it's quite easy to lose organic matter from a soil in a field, especially with continued cultivation, and when no organic matter is being put back into that soil. And so in just summary, I'm just saying soil health is a combination of aspects of the soil, the physics, the chemistry and the biology. And soil tests are important as they provide information on the potential for the growth of that crop and allows potentially adjustments to be made before the crop starts growing with the additions of other nutrients, macronutrients, micronutrients. But the soil structure is also important. The pH of the soil is also important, and these micronutrients may also be important. But combining soil tests with plant tissue tests can help improve the precision of the growing conditions for those crops and hopefully maximize that growth. And I can leave it there. Hopefully that's proved some kind of food for thought for people there without trying to go back too much to basics.
Absolutely, Paul. Cracking statement to finish off as well, isn't it? You know, pH, especially critical, 6.4 for soil, for plants, for humans. It doesn't matter what it is. It's damn important, isn't it? So, yeah, no, fantastic. This is the problem, is that I've got to go and host a session. I wish I was in all of them. Breakout rooms is a great idea until you want to get into all of them at once. But thanks very much for that. So we'll move on now. So we've got Joel live from Canada here, who's going to present us more on a global scale, because we have been a bit behind the curve in UK agriculture now, so we're hopefully catching up a little bit. So Joel's going to take us through some of the work that's been going on around the rest of the world. So we'll hand over to Joel now.
Thank you very much, Chris. Can you see my slides?
Yep, we can. Thanks.
Okay, awesome. All right, great. Thank you very much. And yeah, indeed. Good morning, everybody. It's an early start for me here over in Canada, but okay, I'm going to have a quick look at the idea of plant health, which will definitely overlap a little bit with what we've just heard, which I think is perfect. It'll kind of extend and plug into what we've just heard and also bring a bit of a different perspective on it. The kind of the lens, the framework that I thought I would just kind of briefly talk about plant health this morning would kind of look at these three considerations. First, we'll start looking at it from the design of the agro-ecosystem. How is it that we are planting and managing our crop stands? So first, I'm going to say a few things on this. Then I'm going to do a little bit on the soil biology, just the role of some of the organisms how in which they can also help plant health. And then I'll come back to the nutrient side of things as well. And that's, I will touch a little bit on the refractometer as well at the end here. And that's kind of what we might steer in towards our workshop afterwards. So there's the lens I'm going to talk about. And so I think the first one to really emphasize is this idea of, well, what is the environment that we are designing from the onset? How are we managing plants? Are we managing them, for example, and this would be just one example, but are we managing them under more monocultural stands? Are we looking at more diverse stands? That's from within the field perspective, and then extending that outside of the field. You know, what's happening in the field margins? What's happening in our non-productive areas? Are we using ecology? Are we tapping into those ecological infrastructure in which we can draw from and bring in and utilize at the field level? So I think for me, I think when we shape these discussions about plant health, of course, the wider environment, like always, environments play a massive and significant influence over what crops are doing, be that if it's cold weather, hot weather, be that any of those kind of extremes, temperatures and daylight hours, but also through to the environment in which we intentionally design and manage those plants. So, you know, then things like, okay, zooming in at a field level, these discussions around monocultures versus more diverse stands, and with an element of that diversity comes an element of resilience to that system. Now, of course, here's a very striking example, and of course we can all see a distinct difference in the plants represented in this image. Now, of course, transitioning towards diversity is easier to do in grassland type scenarios. It's a little bit harder to do in a cropping scenario, but still, nonetheless, we see things like intercropping and companion cropping, you know, these types of practices emerging where we're just taking those small steps from 1 to 2 or maybe 3 species within an arable situation, and seeing some of the benefits associated with that. So the point is simply to say that, you know, if our variety and our crop in the monocultural stand, if it is susceptible to a particular disease, then of course that entire stand is going to be susceptible. We've not only have we got the same species, we've got the same variety. So if it is susceptible, the whole field is susceptible. You know, when we have a more diverse stand, we are then diluting that susceptible host out amongst other plants, and therefore just creating a different environment, creating an environment which is a little less conducive to maybe spread of disease or insect attack equally, if the variety was susceptible to the insect attack. So then thinking about diversity, how could we maybe design the system up front with more diversity? And there's 3 ways to think about this. The first one would be using diversity as a tool across time. So this is a temporal dimension. And so here we're just simply talking about rotations. You know, we may still have a monocultural field here in year 1, but we will, through crop rotations, move onwards to a new plant species. So we can use time as a way in which to introduce diversity, which is no doubt the most common tool that we use in production agriculture. But then we could also go to another dimension of diversity and bring in a spatial element. And here I'm talking about that example of, for example, intercropping or companion cropping. So now we have 2 different plants, for example, 2 different plant species. Cereal legume would be the classic combination, that we are now creating a physical barrier between those strips, between those spaces, which then creates physical barriers between the 2 plant species, and therefore can dilute each of those 2 plant species from a pest or insect or disease point of view. And then the final dimension would be using a genetic level of diversity. So here I'm referring to things like mixed cultivars and variety mixes. So now we're going down to that level whereby it may indeed be a monoculture stand of wheat, for example, but within there we may have 2 or 3 or 4 different varieties of that wheat. So we're bringing in a much finer resolution through the genetics there. So that's perhaps the 3 ways in which maybe we think about diversity and how we design the farm system from the onset, which can again be a very important tool influences the outcome of plant health. And bear with me, I'll just quickly explain this one. We won't go into too much detail, but this was just an example of demonstrating some of these kind of dimensions in which we could practically manage. So this was a trial from the Netherlands. This is on potatoes. So here we're looking at, across the bottom, we're looking at time, the days since they intentionally infected the plants with disease. So we're looking at how much time we have after infection. And over here on our vertical axis here, we have the number of the infected leaves, so the intensity of the infection after inoculation. And so you can see the first, the gray bar here is our reference, our control. And you can see that the control here was a monocultural field. Okay, so full field scale, standard monoculture, same variety, standard monoculture. And you can see that indeed after the infection, it was this treatment, the control here, that was the most rapid to ultimately to succumb to disease. We saw the most rapid increase, the rapid spread of that disease throughout the field. So there's our control there. Now, the next treatments that they used was then bringing in an element of that spatial separation. So they've used some strips, 6 meter wide strips, and 6 meter wide breaks, and then similarly again a 3 meter wide strips with 3 meter breaks. So we're bringing in this spatial element here that we kind of talked about, breaking up those strips of potatoes. And so you can see it was the 6 meter that then was the next treatment that kind of succumbed the quickest. We've definitely slowed down over time, slowed down that spread by having physical barriers breaking up those 6 meter strips. But of course, those 6 meter strips are still much bigger and wider than, for example, a 3 meter strip, which becomes a little narrower between each strip. And so the bigger the strip, the 6 meter strip, as you can see there, we're creating a more of a monocultural environment still. So we still saw that more rapid spread there versus a 3 meter strip, which then narrowed those strips. So there was less body of plant material, less width, obviously, less area of plant material in which to spread before there was a break. Okay, so you can see that physical barrier immediately began to slow down that infection. And indeed, the more of those strips or the narrower of those strips, so in the 3 meter example, we saw even further slowing down of those infection rates. Now, the last two treatments here is where they then brought into those potato strips, they brought in variety mixes within those strips. So now we're bringing in the genetic level of diversity. And so over here, you can see again, the 6 meter succumbed quicker than the 3 meter. But once within those 6 and 3 meter strips, once we introduced genetic diversity through variety mixes, you can see that what we are achieving is we're just buying more time across this bottom axis, slowing down the spread, slowing down the infection of that disease after inoculation. And so once again, you can see that the 6 meter variety mix strip succumbed first. And indeed, then when we transition to the final treatment here of 3 meter strips of diverse varieties, you can see that once again, we have bought ourselves even more time. Okay, so there's just a really nice example of how we could use the spatial and genetic diversity. And then, of course, we could do this through time and then bring in that final temporal influence over the rotation as well. So it's just to highlight, again, I think it's a really important nuance in this discussion. We love to focus on plant health from the plant's perspective. We should also widen that lens and think about the environment in which we introduce the plant in the first place. Okay, so a few quick slides then on soil biology, just to say that indeed the biological quality of the soil and the plant biome, these can also be important factors that influence overall plant health. And this was just an interesting slide from a study that was actually looking at the changes in genetic expression in the plant. So we're comparing the changes in genetic expression in the leaf versus down in the roots after an insect attack. And so what you're seeing here is that actually when an insect begins attacking on the plant, there is much greater changes in genetic expression, be that switching certain genes on, switching certain genes off. There's much greater changes down in the roots actually than up in the immediate cells, which are under attack. And this is really signaling to me that, obviously what the plants are doing there is that they begin to, there is a signal, the leaf sends a signal down to the roots to tell the roots that we're under attack up above. and the roots will then change the root exudate profile. They will begin to communicate to the microbes in the soil. They will recruit them, a call for help in order to recruit the soil microbiome to help the plant mount its defenses. So we think about this image, really plant immunity, it's down in the roots. That's where the bulk of plant immunity perhaps happens. And so here's a slide illustrating this. It could be an insect attack, it could be a disease attack, but as soon as the plant is under attack, there will be these signals that go down to the roots, and there will be these distinct changes in those root exudate profile. The plant will release stress signals, certain changes in simple sugars or organic acids, but also many of these other signaling molecules or communication chemicals, in which the plant will then specifically recruit certain microbes, very specific. These can be very specific in many instances, where the microbes from the soil will become more active. They will be recruited by those root exudates. And indeed, those microorganisms that get recruited after infection or attack, they then essentially help that plant mount its defenses. So they might increase nutrient uptake for the plant, but they also might release very specific microbial chemicals, biochemicals, microbial compounds that the plant can also use to again mount its defenses, whereby those defenses can be then systemically translated throughout the plant. So these could be antifungal type compounds or cellular structural compounds to build a barrier, or equally other more bitter defense chemicals that might deter the insect pest, for example. So just to highlight that then there is a role of the soil biology in helping the plant drive its immune system. And this is a study that was looking at exactly that, as the title suggests, that the initial soil microbiome composition and function predetermine future plant health. So here we explored how variation in the initial soil microbiome, the initial health or functionality of the soil biology, ultimately predicted the future disease outcomes. They found that the composition and the functioning of that initial soil microbiome predetermined whether plants survived or succumbed to disease. Surviving plant biomes were associated with very specific taxa. There were certain microbes that were being recruited, Pseudomonas and Bacillus in this example, and particularly a high abundance of genes that encoded for the production of these antimicrobial compounds, these defense chemicals and things that then suppress the disease, just as an example. So you can see by, indeed, let's be honest, the world of soil microbiology is a big black box. There are many unknowns, many knowledge gaps, but we're beginning to piece those together. And this was just an interesting study. Looking at that, we can see that ultimately plants that grew on to be healthy, there were distinct differences in the soil microbiome composition from healthy versus plants that went on to become diseased. So it appears that the biological quality of the soil does indeed play an important role. And so lastly, then, just to say a few things on nutrition, and that'll lead us into our workshop, is just to say that then the role of minerals, essential macro and micro minerals, which we heard nicely from our former two speakers, those minerals are catalysts in driving photosynthesis and driving plant growth, plant biomass, etc. And it's simply just to say that if we think about the role of minerals acting as these catalysts or part of enzyme systems, is that really they are the key that enables photosynthesis to function. Okay, breathing in carbon dioxide, taking up water, building sugar, building glucose, that building block of the plant body. But we can't do that without the essential minerals. And for example, manganese, nitrogen, magnesium, iron, certain ones that have very direct roles in that initial photosynthetic process to take carbon dioxide and turn it into sugar. But then the plant takes that glucose as a building block, and then it stitches it together, builds a whole host of other plant compounds. So it takes that simple building block and turns it into more complex sugars, complex carbohydrates, linking in nitrogen, sulfur, building amino acids, proteins, other fats, oils, other hormones, plant growth regulating compounds, smells and scents, volatile organic compounds, other phytonutrients, and these defense chemicals that we were just talking about. But again, the key is the plant requires minerals, essential macro and micronutrients, again act as catalysts, as part of these enzyme systems that then help facilitate this conversion from building block into all of these other plant compounds. So this is why the plant nutrient status is really important, because it is those minerals. We can't synthesize these things without the presence of those minerals. So if we were to think about this process of plant growth and photosynthesis from a insect and disease kind of resistance point of view, the process is the same. We have to drive photosynthesis. First, we need the building block, but then the plant, again, using specific minerals to convert that building block, the plant can synthesize these anti-feedant, bitter compounds, anti-herbivory type compounds, again, cell strengtheners, deterrent chemicals, so again, volatile compounds that attract in beneficials or repel the pest, and all of these various kind of protective and defense chemicals inside, and those root exudates that we talked about earlier to recruit the beneficial microbes from the soil. So again, it comes back to photosynthesis and the nutritional status of the plant in driving this process. Same deal with disease. Again, we take the building block. Again, we need the macro and micro minerals that then can turn that building block into different sets of defense chemicals, antimicrobial, antibiotic type compounds, again, physical barriers or cell strengtheners, and some other defense chemicals and things again. Okay, so it's the same principle. Ultimately, we've got to optimize the plant nutrient status, be that for production or be that for plant health and plant resistance, insect and disease resistance. Now, the tool, a kind of a crude and beginner's tool, we're here in a beginner's session today, a tool that we can kind of use is the refractometer. And like Steve also mentioned, it has some strengths and weaknesses. The environmental influences become a really important factor. It's not a perfect tool. It's a little bit of a blunt tool, but it's better than nothing. It's still a tool in which we can use to certainly compare good, as relative treatments, compare good and bad areas, and also just monitor these things over time. So when we talk about the refractometer, what we're doing is measuring photosynthesis or the products of photosynthesis. We're measuring these products back here. We're measuring not just the sugars, but we're also measuring all of these things that are dissolved in the plant sap, the carbohydrates, the amino acids, the proteins, any of the defense chemicals in any of these products of photosynthesis is what we are measuring with a refractometer, all these compounds here. And of course, we can't synthesize those without the minerals. So indirectly, we're using the tool to measure the products of photosynthesis, but make indirect assessments of the plant nutrient status, because we can't have these without those. Okay, so that's what we're doing with the refractometer. It therefore gives us this overall gauge of plant health, plant quality, plant immunity, but indeed there are many environmental variables that influence this, and some of this conversation will carry on perhaps through into the workshop. When we use a Brix meter, we are extracting some plant sap, taking a leaf, crushing the leaf with a garlic crusher, for example, extracting some sap, putting a drop of that sap on this meter to take a reading. This is kind of what we see, and the goal here is the more of those products of photosynthesis, the density of those products, the more sugars, the more complex carbohydrates and things that we have in that plant sap, the higher the Brix reading will be. Okay, so we can see on the left side here, we have a very low reading of 4, and that line is very crisp. It's a very sharp line between the blue and the yellow. So this is a low Brix reading plus a very crisp line. And over here on the right-hand side, we have a very high Brix reading plus a blurry line. Okay, so what you're chasing is a high and a blurry line as possible. This is your best indication of plant health. And the reason that is in summary, just to kind of close off this section here, is that this is kind of what we're tapping into. If you're seeing a very crisp line, it really basically means you're at a foundational level of plant photosynthesis. You're kind of at this first step where you're only really producing more of the smaller, simpler sugars. It takes more nutrients, additional nutrient plant status, to then take that building block and convert it into all these more complex plant compounds. And so when we then have additional nutritional supply to facilitate the second phase, the second kind of stage of plant metabolism, and we then synthesize more of these complex compounds, complex sugars, more of the proteins, particular some of the fats and lipids, these more structurally complex compounds, they are the ones, when they are present in the plant sap, that then we see more of this blurry line. Okay, so in summary, it becomes just a basic tool. There is a lot of environmental noise for sure, but the refractometer becomes a basic tool in which we could look at two plants or do some trial work, like Steve was talking about, and try and observe some of these differences as a crude tool in field, rapid assessment of how well the plants are photosynthesizing, and therefore potentially how well the nutrient, how optimized that nutrient status is for them to then fuel that immune response if they get attacked. How much nutrients do they have to produce those defense chemicals against the insect or the disease if they are under attack? Okay, so that's my view on plant health in a nutshell. Ecology, biology, and nutrition.
A very quick run through it as well, Joel. So we've just got, on my screen here, we've got a little sample that was very difficult to take a picture through a Brix meter, but we did manage it with David there early season. That was the March reading through the refractometer, which, for a £20 instrument off of Amazon, it's a pretty quick, easy way to quickly judge it. And I think we're getting used to now that there is a bit of a toolkit you need, whereas it's the pH meter, you've got the Brix meter there, which then, depending what it tells you, if it's a clear line, you've got the calcium deficiency, and then you move on to your potassium meter. If it's high potassium, you've got low mag. If you've got low potassium, you've got high mag, and it's just trying to balance these things. So from a £20 meter, you can actually do quite a lot from it. So we'll maybe go back to our facilitators here. Hopefully that was an interesting session. So Johnny, do you want to kick us off with a wee recap of your session?
Sure. So we started off talking about the transition that Joel was talking about in his presentation, and the first steps that you can take and the length of time that it might take, and that there are steps you can do embracing the regenerative agricultural principles in the first year, but you're looking at a 3 to 5 year time period before you can actually get a meaningful defenses in your plants that are coming from the soil. We then had lots of discussion around Brix meters and how to use them, how to interpret the results from them, and that we came to the conclusion that doing a small number of fields on your farm and doing them regularly is much better than doing a kind of haphazard all across the farm, looking at every field in every year. No, just pick a handful and do them regularly, and that the Brix meters, the individual results are not as important as the trends over time and over space. So looking at them over the field and over the handful of fields that you're doing at that time, and looking at how those results change over the season that you're taking the results. We briefly touched on farm tech, but I won't go any further with that. And that in terms of nutrition, much better to piggyback nutrition into your fungicide program, and you should start to improve things. Yeah,
absolutely fantastic. Thanks very much for the recap. I think we're very similar in terms of how is this applicable on farm. David, we'll hand over to you. Well, I'm looking at you, you're only there, but yeah, we'll hand over to you next as well.
Yeah, thanks, Chris. Yeah, we had an interesting question. Dave Bell set us off with a question about carbon, and we got into discussion about how carbon, essentially, Paul was highlighting, is essentially a gross measure of what's in the soil, and we touched on the fact that it's kind of a bit like looking at, it's a proxy measure for organic matter, whereas actually organic matter is more a measure of the kind of biological side of what's in the soil, is my understanding, with a view to looking at soil health. We talked about Thomas, who was in our group, had a CO2 burst meter, which I got quite excited about until he told us the price of it, and we thought, well, we'll just get Chris to open his wallet and sort that one out at some point potentially. But it certainly looked like, when you see the technology involved, you thought actually it might be a useful tool that could be shared by a few growers possibly, and actually just keep a track on your soil health if you're that far into that type of thing. Yeah, and that's kind of what most of our discussion kind of based around. And then how do we go forwards? How do we, farmers doing work, sharing work together? You know, you can achieve a lot more if with 4 or 5 farmers trying something in any one year. You potentially get 4 results in 1 year, and we can learn from that. And the recognition that actually there are scientists working in the background, but most farmers are doing some form of trial work on their farm, be it with anecdotal results, whether it's trying a variety mix, different species, so on and so forth. And it's just the need perhaps to collate some of this information and take steps forward as an industry, and how we turn the science behind soil health into practical realities on the ground. We all know that we should be applying lots of dung and maybe have some livestock on our farm, etc. But in a practical world, that's not always an option for many producers. So it's how do we bridge those gaps along the way?
Yep, fantastic. It just, as always, it seems time's the limiting factor, isn't it? Even in these sessions, you know, we were just getting into our discussion, which was fairly kicking off. And again, our group with Steve there was, you know, how do we take what the Balbirnie team here are doing in terms of John and David and, you know, his team of guys on the ground, and how is that relatable to the rest of the farms? And, you know, questions, how does the agronomist pick this up? Can he be in a crop every week? You know, versus a farmer doing it himself at these timings, and what are they looking for, and how are you going to react to that? And I suppose we came to the rough conclusion, if I'm allowed to use that word, that you know, the farmer knows his ground and his soil and the parts that are good and bad. And interesting here, and Johnny and Joel, that you know, you need to look at the bits that you specifically want to look at. Are you going to change these? Are you trying to change these? Is that your agronomist's job? Is that your job? Whose job is that? And I suppose these are all very, very relevant questions in terms of it. And then, you know, we briefly touched on, you know, the drivers for this and the loss of active ingredients that's always coming at us, you know, with Bravo going, the chlorothalonil, the epoxyconazole, the ones that are coming at us, whether it be glyphosate or prothioconazole. You know, what's next? Where are we going with these products that we're leaving? How long are we going to have these for? You know, where is the next step? And I suppose that's where this is a farmer-led, farmer-driven trial. This is where the farmers are wanting to go, and that's what's really pushing this forward. And everything else really follows from that. You know, in terms of the foliage in the ground, well, we'll just drill a seed. It's only a seed you're putting in the ground, so that's where the direct drilling comes from. It's a tool in the toolbox. It's not that that's driving it. It's the whole system. So I think, look, we've a little bit of time here if anyone's wanting a couple of questions. So I think there's one there from Emily to Joel. In terms of looking at Brix meter reading trends, what other data should we be keeping in a record of so that we can interpret those trends over multiple years, which I think we were getting to as well, Steve, weren't we?
Yeah, look, I'll jump in. I'll quickly jump in. I actually just finished typing to you, Emily. No, here I am anyway. All I just finished typing there was to say that, yeah, it's of course, it's a really good question, and we had a little bit of this discussion in our workshop. But just to say, I think that I would add a little notes column to that, rather than just the Brix reading, the number itself, and whether or not it's a crisp or a blurry line. I would just add a little notes section to say, okay, well, what was the weather of that time? Acknowledging that the climatic influences play a big role. You know, was it a sunny day? Was it a cloudy day? Has it been dry? Has there been recent moist rainfall? Any of those kind of additional notes on the environmental factors, just to help contextualize the reading itself, is probably another few other pieces of information I would add. That way, in the future, when you are looking back at the number and the numbers over time and those trends, you're also perhaps bringing in some of the just more, you know, subjective and qualitative notes around what the number actually was. So I would include things like that. But then beyond that, sure, other meters like Chris, you mentioned, maybe the chlorophyll meter or the sap pH meter, etc. Just maybe some other in-field meters that bring also some other numbers to build a bigger body of data that you could interpret. But I would also say that I think, of course, certainly a full tissue analysis or sap analysis to assess, the full spectrum of macro and micro minerals, I would definitely also assess. To get that full diagnostic of the full nutrient status, I would definitely be using those tools as well.
Absolutely. And Emily Grant there, does the soil CO2 burst measure rely on re-wetting dry soil? And if so, does it only give a brief measure of the microbes that respond quickly after a dry period? We usually don't have very dry to wet, so are they useful tests for Scotland, applicably?
Yes, from what I understand from our discussion, the meter that was being shown was working on dried soils. There are ones that will work on wet soils, but take a bit longer to get your, work over a longer period of time. So yes, these have been used on Scottish soils, and we have got results for them as well.
We'll have to draw our session to a close there. Hopefully that went well for everybody.